Hey guys, and welcome to my second lecture. So in my earlier session we just discussed about the type of microscope, that is a material characterization. So it deals about the various characterization are there. In specific we are going to speak about the scanning electron microscope, and we know that how the scanning electron microscope, first of all we need to know how does it work. Generally scanning electron microscope is the type, okay, it is the type of electron microscopy that images on the sample surface. It can be obtained by using the scanning by passing the high energy beam of the electrons. So generally this scanning can done by through the electron gun. It can produce the high amount of intensity electron which can be strike over the condenser lens. So this condenser lens will absorb the light rays, and it can try to converge the deviated light rays into converger one. So once the convergence has been done, that following electrons wavelength which has been scattered throughout their rays, and it can, by using the deflector coils, this rays can be again getting deviated to improve the intensity of the beam which can strike over the object. Okay, it can producing the signals that contain the information regarding the sample surface topography, and there are some other properties which helps to determine the surface characteristics only by through the backscattered electron detector. So this image whatever you seen here, this electrons has been getting deflected, and with respect to that the X-ray detector has been used, and by using this objective lens it can be getting deviated over the sample, and accordingly that sample has been displayed with the help of the vacuum pump. Okay, so this is why we are getting the clear images. For example, let us take a right side image which shown that number of layers over a single sample. So by pausing the rays, when the deflector rays has been keep on striking over this sample, it can able to trace it out what kind of layers and how many layers are there. Accordingly we can get a result. So that is what the primary purpose. Okay, so let me see the one by one about this scanning electron microscope. So what actually it does. So actually this scanning electron microscope, here the images what you seen, here these are the three different kind of images which can be taken by using this same, that is a scanning electron microscopy. Okay, here the metal matrix has been taken for the consideration. These are the surfaces over. Actually this sample has been made with the help of the casting. So we know that when we made the sample over the casting, we have to analyze what are the metal matrices has been mixed in that particular sample. Okay, so this images, when the electron beam strike over the surface, it can trace it out the topography of this image. Okay, and let's take about the second image. Here this is about their a plant cell which has been taken for the consideration. Okay, it is a pollen. These three different images here, this is one more deer tick. Okay, the deer tick means the inside the deer tick what is the kind of structures has been find out, and how it can be traced. So by using these three different kind of images, we can study about actual surface characteristics of the particular sample which we are taken for the analysis. So these three different images having the secondary electrons, that is a low energy electrons from the surface of the material, it can be backscattered the electrons more from the bulk, and the X-rays from the near surface region. So that is why we are getting this kind of simple rays which are making through the metal corrosion layer, and with respect to the pollen has been we are finding out their high intensity beams. That is why we are using the same for analyzing the surface characteristics. Okay, and here it was shown there is a same X-ray analysis. So here it shows what kind of chemical composition which has been present over the surfaces by the rays. Whenever it attains the peak, this peak shows the huge amount of electron get reflected, and whenever the peak is very low, it shows the electron get deflected. That is, there is no reflection will taken place. That is why the composition has been getting changed. It has been mentioned by using the chemical names. Okay, and we will continue next session. Thank you.